Hello, my dear friends. Welcome to the presentation. So let me start by saying a few words about my own background. I'm Dr. Indrajeet and I'm working as an assistant professor in Department of Microbiology in KTHM College. So today is the second part of the cell cycle seminar. And before we move on to the second part, I would like to give you a brief recap of what we have learned during the last seminar. So during the last seminar, we have seen a brief overview of cell cycle phases. So the cell cycle consists of three different phases, that is G1 phase, S phase, and G2 phase, which together constitute an interphase. And after interphase, it's a phase called mitosis, in which the duplicated chromosome gets segregated into two daughter nuclei. And we have also learned about different phases of mitosis, which are prophase, metaphase, anaphase, telophase, and cytokinases. And after that, we have also discussed about range of different techniques which we will use in the laboratory to identify different phases during the cell cycle progression. And I also elaborated on the cyclin and the CDK system, which are the central components of the cell cycle regulatory machinery. And finally, I have concluded the first part of the seminar by explaining you the transition which happens during G1 phase that is typically mediated by two important cyclins, that is cyclin D and cyclin E. So for the next few minutes, I have divided my presentation in the following points. So first, we will start with the cell cycle transition, which specifically happens during S phase of the cell cycle. Then we'll also learn about the transition which takes place during mitosis, and followed by that, we will look into the details of the centrosome duplication. So these centrosomes are important organelles which radiate microtubules, and we will see how they exactly duplicate. And following that, we will also learn about checkpoints, which are nothing but the surveillance systems. And these checkpoints are of three different types, that is P53 mediated checkpoint, an ATM and ATR mediated checkpoint, and finally we will look into the spindle checkpoint, which is mediated by anaphase promoting complex. And then we'll also check for the brief overview of meiosis, and we'll look about the comparison between meiosis and mitosis. And finally, I would like to conclude the second part and the final part of this seminar by explaining you if there is a defect in the cell cycle component and how exactly it results in the tumorigenesis. So let's begin with the brief overview of the cell cycle transition which takes place during S phase of the cell cycle. So during the S phase of the cell cycle, chromosome duplication happens. So this chromosome replication typically takes place as one of the important sites on the genome, that is origin of replication. So on the origin of replication, the origin recognition complex protein binds. And there is also one catalytic protein, which is CDC6, which is loaded onto these sites. And another important protein which is required to initiate the replication is helicase, which we called it as an MCM helicase. So this MCM helicase in G1 phase is also loaded near to the origin of replication sites. So during G1, this results in an inactive pre-replicative complex which assembles at the site of origin of replication. So as soon as the cells enter into the S phase of the cell cycle, the cyclin and the CDK, which are specific for the S phase, gets activated. And that results in the activation of some of the initiator proteins which are important to initiate the process of DNA replication. In addition, this cyclin and the CDK complex, which are specific for the S phase, also inactivate one of the ubiquitin ligase that we called as anaphase promoting complex. So as a result of this inactivation, there is activation of the DDK kinase. And these DDK kinase are important in order to phosphorylate MCM proteins. So the phosphorylation catalyzed by this DDK kinase, which in turn activates the MCM helicase proteins. So the activation of MCM helicase results in the bidirectional replication, and then this MCM helicase starts unwinding DNA in a bidirectional manner. And then at the end of the G2 phase, there is completion of replication which happens. So as the replication is one of the very vital process, and the cell has to ensure that the genome segments replicate only once during one cell cycle. And this is in turn governed by the CDK kinases. So the high activity of the CDK kinases during SG2 and M phase results in the phosphorylation of the origin recognition complex protein, and this phosphorylation of the ORC proteins results in inhibition of the MCM helicases. So the MCM helicase cannot load onto the origin of replication and then cannot initiate replication during S and the G2 phase. So thus, once an origin becomes active during S phase, replication at that origin cannot initiate again until the cell passes through mitosis and enters in the G1 phase of the next cell cycle. So as the replication happens, the sister chromatids are formed. And there are important set of proteins which maintains the architecture of these sister chromatids. And these are nothing but the cohesin proteins. So these cohesin protein are SMC family of proteins. So the SMC family means structural maintenance of chromosomes. And the structural maintenance of chromosomes, the function of this family is to maintain the chromosome architecture, structure, as well as the genome integrity. So these cohesin molecules have two important proteins, that is SMC1 and SMC3. So the SMC1 and the SMC3 has the ATPase domain on the C and the N terminal side, where it also has the hinge region in the central part of the protein. And there are some of the additional components which are SCC3 protein and SCC1, which are important to bridge to SMC1 and SMC3 proteins. That results in the functional ATPase domain of cohesin. And without this ATPase function, the cohesin cannot load onto the chromatid. So once the replication happens, immediately the sister chromatids are encircled with the help of cohesin molecules. And these cohesin molecules are actually located at the site of centromeres as well as across the length of the chromosome. So during metaphase, normally this cohesin ring has to be broken in order to activate the segregation of the sister chromatids. So let's see what happens during cell cycle transition, and that too specifically in the mitotic phase. So the first and the foremost thing which happens during mitosis is activation of the mitotic cyclin CDK complex. And this mitotic cyclin CDK complex is cyclin B and the CDK1. But before mitosis, this cyclin B and the CDK1 has been kept inactive. And this is with the help of the kinase which we call as a V1 kinase. So the V1 kinase actually phosphorylates the CDK1 at one of the inhibitory residues, that is threonine 14 and tyrosine 15. So the phosphorylation at these residues results in the inactivation of the cyclin B and the CDK1 complex. But as the cell enters in the mitosis, there is the activation which takes place by dephosphorylation mechanism. And one important protein which catalyzes this dephosphorylation is CDC25 phosphatase. This CDC25 phosphatase removes these inhibitory phosphate molecules, that is threonine 14 and tyrosine 15, and results in the activation of this mitotic cyclin B and CDK1 protein. So once this gets activated, it carries out variety of processes which are important to initiate as well as to complete the mitotic process. So during metaphase, this cyclin B and the CDK1 protein are actually cleaved by anaphase promoting complex. So this anaphase promoting complex is a ubiquitin ligase which ubiquitinates cyclin B residues, and that results in the degradation of these proteins. So once the cyclin B and the CDK1 activates, we will see what kind of processes it contributes towards. So cyclin B and the CDK1 and Aurora1 and polo-like kinase, they all of the three together function in a positive feedback loop at the onset of mitotic phases. So they activate each other. So once these three kinases get activated, they carry out variety of processes which are required to initiate the mitosis. So the first thing which happens is chromosome condensation. So this chromosome condensation happens after phosphorylation of condensins and cohesins. Then the second thing which happens is the nuclear envelope breakdown. And so these three kinases catalyze phosphorylation of the lamins and one of the important components, that is the nuclear pore complexes and the nuclear membrane proteins. And this results in the nuclear membrane disintegration, and finally it ends down in breaking of nuclear envelope. And the third thing which happens is the fragmentation of the Golgi apparatus. So the phosphorylation of Golgi apparatus takes place by the help of these kinases, and that results in complete disintegration of the Golgi apparatus. And the final and one of the very important thing happens is the spindle formation. The spindle formation happens with the help of Kinetochore centromeres, and the microtubule associated proteins. And these are in turn get phosphorylated with the help of these kinases. So let's look at what happens with respect to cohesins and the condensing molecules. So as the cell passes through the replication and enters in the G2 phase, the cohesin molecules encircles the sister-chromatids. And that encircling happens across the length of the chromosome as well as at the centromeric site. But as soon as the cell enters in the mitosis, there is activation of the mitotic cyclins, which are CDK1, cyclin B, which operates in a feedback loop in order to activate the other kinases which are important. Those are Aurora B and polo-like kinases. So the first thing which is carried out is by CDK1 and cyclin B, which phosphorylate cohesin molecules. So the phosphorylation of cohesin molecules across the length of the chromosome results in evacuation of cohesin from the chromosome arms. But chromosomes still exist at the centromeric sites, as you can see here. And Aurora B and polo-like kinase in turn phosphorylate condensins, so the condensin accumulates at the base of the chromatin loop. And this in turn results in the chromatin condensation, which you can see during the metaphase as one of the duplicated chromosomes. So this chromatin condensation is one of the very vital process. And this happens with the help of histones. On histones, the DNA molecules wrap itself and forms a bead on string model. And that results in the formation of the chromatin. And then chromatin fiber folds onto itself, which results in the formation of the nucleosomes. And then these 30 nanometer nucleosome fibers also fold around itself with the help of cohesin and condensing molecules, and that results in the severe compaction. And that you can visualize as during the sister-chromatid. So why it is important to compact this DNA? It's because after replication, the sister-chromatid have been disentangled from each other and lie side by side. Thus, the sister-chromatid can easily separate when the mitotic apparatus begins pulling them apart. And the second important thing why the compaction is important is because compaction of chromosomes protects relatively fragile DNA molecules from being broken as they are pulled apart to separate into two daughter cells. So the second thing which happens during mitosis with the help of kinases is the nuclear envelope breakdown. So this nuclear envelope consists of wide range of proteins. Those are nuclear membrane proteins, nuclear pore complexes, as well as nuclear lamina. So once the cell enters in the mitosis, as we already know that the CDK1 and cyclin B gets activated, and this activation of this cyclin CDK complex results in the phosphorylation of the lamins. As you can see here, the lamins are present just beneath the nuclear membrane, and they act as a scaffold or to support the nuclear membranes. So the phosphorylation with the help of CDK1 and cyclin B results in their disintegration and results in the formation of the lamin dimers. And finally, this lamin completely breaks down, as you can see here, and that results in the complete nuclear envelope breakdown. And one of the important things which happens during metaphase to anaphase transition is activation of anaphase promoting complex. So during metaphase to anaphase transition, the cyclin B and the CDK1 has to be broken down. So this is catalyzed by one of the important family of ubiquitin ligases, that is anaphase promoting complex. So normally at the onset of mitosis, you can see the metaphase chromosomes which are exactly bound at the centromeric position by these cohesin molecules. So this cohesin molecule has to be broken down in order to segregate them into the sister-chromatid, which happens during metaphase to anaphase transition. So at the onset of mitosis, anaphase promoting complex is present in an inactive form. So for its activation, it requires one of the catalytic subunit, that is CDC20 protein. And this CDC20 protein in turn binds to anaphase promoting complex. And so binding of this CDC20 protein results in an activation because of the huge conformational change in the anaphase promoting complex protein. So once it's get activated, it carry out ubiquitination of securin protein. So what does securin functions? So securin sequester the separase protease. So this separase is very important in order to cleave the cohesin ring. So the ubiquitination of securin by anaphase promoting complex, that is nothing but a ubiquitin ligase, results in a complete degradation of the securin protein, and that releases the active separase protease. So the active separate protease then in turn cleaves the cohesin which is present or which is bridging the two sister chromatids. And because of this cleavage, that results in the segregation of the sister chromatid into the anaphase. And one important thing which is catalyzed by anaphase promoting complex is inactivation of mitotic cyclin and the CDK in order to progress it from metaphase to anaphase transition. And that is in turn catalyzed by binding of anaphase promoting complex with CDC20 protein that results in activation of this anaphase promoting complex, and this activation in turn catalyzes the ubiquitination of the cyclin molecules. So the ubiquitination of this cyclin molecule results in the degradation and finally results in an inactive CDK protein. So because of this, there is degradation of this M-phase cyclins, and that finally results in a successful metaphase to anaphase transition. You can also visualize this transition under a microscope. So here we can see that the chromosomes, which are blue in color, and the microtubules, which are stained with gold label antibodies. And this we can observe under a normal light microscope. And you can see here on the left, that is on the metaphase, the chromosomes are equatorially aligned. And after the metaphase to anaphase transition, this results in the segregation of the sister chromatid into the two daughter nuclei. So let's see the third important point of this seminar is the centrosome duplication. So centrosomes are very important in order to radiate the microtubules. And this centrosome are actually present around itself is a green perionuclear matrix. The centrosomes has a pair of centrioles, and at certain point in G1, these two centrioles of the pair separate by few micrometers. And during S phase, as the DNA gets replicated, the centriole also begins to replicate itself. And that to a new centriole is formed at right angles to the origins of centrioles. Then the elongation of daughter centriole usually completed in the G2 phase of the cell cycle. And this duplication of centriole is very similar to that of DNA duplication. That is, it's a semi-conservative mode of duplication. And during mitosis, the two centriole pair remains very close to each other in a central centrosomal complex until the beginning of mitosis. And here, the complex splits into two, and the two daughter centrosomes begin to separate. So each of the daughter centrosomes now nucleates its own radial array of microtubules, and we called it as an aster, and mainly from the mother centriole. So there are different types of metaphase spindles which are formed from the centrosomes. The first is the kinetochore microtubule that attach to the chromosomes. The seconds are the interpolar microtubules that overlap in the center of the cell. And the third are the astral microtubules that radiate from the centrosome to the cell periphery. So with this video, I would also like to elaborate the kinetochore and how exactly it plays a role in segregation of this sister-chromatid.
This visualization isolates a single chromosome at mitosis. The chromosome contains two identical copies of DNA. Each DNA molecule is packaged up into one of the sausage-shaped chromatids. Sticking out from either side are microtubule fibers from the mitotic spindle used for orienting and guiding the chromosomes to their correct positions. The red region is the Kinetochore, which works as the interface between the microtubules and the chromosome. The Kinetochore is one of the largest and most complex molecular mechanisms inside living cells, with multiple functions to perform. The kinetochore is central to the movement of the chromosomes and is able to hang on to the dynamic ends of the microtubules. It is also a mechanical tension sensing system, which is able to feel if everything is correctly attached and positioned. When all the kinetochores are ready, the cell transitions into the anaphase stage, with the chromatids pulled apart to the opposite poles of the cell.
So let's look in detail about the checkpoints. So the checkpoints are nothing but a surveillance mechanism that ensures a unidirectional flow of cell cycle progression, and that too in a proper manner. These checkpoints are in turn activated mostly by DNA damage during the cell, and that is a p53 mediated checkpoint and ATM and ATR mediated checkpoint, which are activated by DNA damage. And third, the spindle checkpoint, which is regulated by anaphase promoting complex, and that is in turn activated by misalignment of the chromosome at the equator and their binding with respect to the kinetochore and the respective microtubules. So let's see what happens during the checkpoints. So this is a general overview of different checkpoints which operate during the cell cycle progression. So G1 checkpoint and the G2 checkpoint operate because of the DNA damage, whereas the mitotic checkpoint happens because of the misalignment of the chromosome at the equator. So let's see what happens during DNA damage checkpoints. So our cell is regularly attacked with a lot of exogenous environmental factors or some of the endogenous cellular events which can lead to DNA damage. So this DNA damage can be either of two types. It can result in a break in a single strand of the DNA, and that results in activation of ATR kinase. So these single-stranded breaks are in turn sensed by some of the sensor protein that initiates a signaling cascade, and that we called as a DNA damage response, which results in activation of the central kinases. And with respect to single-stranded breaks, this central kinase is ATR, that we called as ataxia-telangiectasia related protein. And the activation of this ATR kinase results in activation of the CHEK1 kinase. And on the other hand, there are different types of breaks, and one of the deleterious or the most catastrophic break is the double-stranded break. So these double-stranded breaks are sensed by the sensor proteins, which in turn activates a signaling cascade and then in turn leads to activation of ATM kinase, which is a central kinase. And the long form of ATM kinase is ataxia telangiectasia mutated. So this ATM kinase, once it gets activated, it results in the phosphorylation of the CHEK2 protein. So this activated CHEK1 and CHEK2 in turn inhibit CDC25 protein. And as we already seen, that the CDC25 is a phosphatase that is important to remove the inhibitory phosphorylation and is very important to activate the cyclin and the CDK kinases. And here, the CDKs are CDK1 and CDK2. And the inhibition of the CDC25 by this CHEK1 and CHEK2 protein in turn results in inhibition of the cell cycle progression. In addition, there is also an activation of a central tumor suppressor protein that is p53. And let's see how exactly it gets activated. So in terms of double-stranded breaks, which are normally formed because of the radiations or different endogenous cellular events, leads to activation of ATM and ATR kinases. So this ATM and ATR kinase in turn activates the CHEK1 and CHEK2 kinases. And this CHEK1 and CHEK2 kinases results in phosphorylation of the MDM2 protein. So what is this MDM2 protein? This MDM2 protein is an inhibitor of p53 protein. So in a physiological state, this p53 is bound by one of the inhibitor, that is MDM2. So in order to activate p53, the MDM2 proteins needs to be removed. And this is taken care by the CHEK1 and CHEK2, which phosphorylate this MDM2. So the degradation of MDM2 results in an activation of p53. And p53 is one of the very important and crucial activator of DNA damage response. So this p53 is a transcription factor. It translocates into the nucleus and then binds to variety of promoter regions. So it activates a wide range of genes, and one of the very important gene is p21. So as the p21 mRNA is formed, that results in the translation, and that leads to formation of p21 protein. So this p21 protein in turn binds to the G1S cyclin as well as S-phase cyclin CDK complex. So we have seen this mechanism in the first part of my seminar, that this p21 is a CYP inhibitor family which binds to the CDK and that sequesters CDK cyclin and their active site from functioning. So tightly bound p21 results in a conformational change in the cyclin CDK complex, and that leads to its inactivation. So as we already know, once the cyclin CDKs are inactive, then it leads to complete inhibition of its transition. And this is how p53 plays a very important role in cell cycle transition. So the third checkpoint we will look into details is the spindle assembly checkpoint. And this spindle assembly checkpoint is mediated by Anaphase promoting complex. So this checkpoint is very sensitive. Even a single misalignment of a chromosome can lead to activation. So let's see what happens when the kinetochore remains unattached at the centromeric site on a chromosome. That results in an activation of MCC, that is mitotic checkpoint complex. This mitotic checkpoint complex consisting of different proteins that includes BUB-R1, MAT2, BUF3 and CDC20. And as we already seen in some of my previous slides, that CDC20 is an activator of Anaphase promoting complex. So because of this unattached kinetochore, that results in activation of this MCC complex, and that in turn leads to inactivation of Anaphase promoting complex by sequestering its one of the important catalytic subunit, that is CDC20. On the other hand, if there is a proper alignment happens, that results in the release of CDC20, which activate Anaphase promoting complex, and the activation of this Anaphase promoting complex works in two different directions. So the first and the foremost thing which happens is the degradation of the cyclin and the CDK proteins. So there is ubiquitination of cyclin B which happens, and that leads to its degradation, and that results in activation of mitotic cyclin CDK complex. On the second side, which happens once the anaphase promoting complex gets activated, it results in the ubiquitination of the securin protein. And as we have seen in the previous slide, that the securin is involved in sequestration of the separase protease. So once the degradation of the securin is initiated, then there is a release of this separase protease. And the separase protease, as usual, it functions at the centromeric sites and it removes the cohesin, which is tightly holding, or which acts as a glue which holds two sister chromatids together. So as a result of the cohesin cleavage, there is a segregation of sister chromatid into two daughter nuclei. And that results in a successful metaphase to anaphase transition. So nextly, I would like to give you a brief overview of meiosis, and we'll also compare it with the mitosis. So meiosis is a form of nuclear division in which a single round of chromosome duplication, which specifically happen in meiotic S phase, is followed by 2 rounds of chromosome segregation. So this is a pictorial representation where a fate of a single homologous chromosome is followed during meiosis as well as in the mitosis. So during meiotic S phase, on the other hand, in a mitotic S phase, the DNA replication happens. And as we can see here, there is red and the gray color. Whereas in the meiosis 1 is completely different than the mitosis. So in meiosis 1 consists of different phases which are similar to mitosis, but they are chemically quite different. They include prophase 1, metaphase 1, anaphase 1, and the telophase 1. So in prophase 1 of the meiosis 1, the sister chromatid cohesion happens. And there is a crossover which happens between two homologous chromosomes. And then there is a segregation of this homologous chromosome which specifically happens during anaphase 1. And then the next steps are almost similar to that of metaphase of mitosis, where the homologous chromosomes which are segregated from meiosis 1 gets aligned at the equator, and then the segregation is initiated. And following segregation in meiosis, there are formation of 4 haploid, and that too a genetically different haploid nuclei are formed. On the other hand, there are 2 diploid nuclei in mitosis are formed which are genetically identical. So we will also look for the comparative differences between meiosis and mitotic chromosome alignment. So in case of meiosis, the homologs are paired side by side to each other. And this side-by-side pairing result in attachment of the sister kinetochore, which we can see here, and that too only from one mitotic pole. And that results in the segregation which specifically happens in the anaphase. So the homologous chromosome gets segregated into 2 daughter nuclei. Whereas the metaphase 2 of meiosis is almost similar to that of mitosis, where the homologous chromosomes are equatorially aligned and they are bound by the spindle microtubules from 2 different folds, and that we call as a bipolar spindles. And that results in the segregation in anaphase 2 as well as the anaphase of the mitosis. And end product of meiosis is again 4 genetically different haploid nuclei, whereas in mitosis, there are diploid daughter nuclei which are genetically identical. So finally, I would like to end this cell cycle seminar by explaining you the importance of the cell cycle components. And if there is any defect in this cell cycle component, that might result in initiation of tumorigenesis. So one of the important cell cycle components which are crucial are nothing but retinoblastoma proteins. And we have seen in the first part of my seminar that the retinoblastoma actually sequester a crucial transcription factor, that is E2F. And this E2F sequestration result in the inhibition from the G1 to S-phase transition. So following a restrictive point, the CDK4 and cyclin D1 gets activated. And as a result, it phosphorylates a retinoblastoma protein. And the retinoblastoma protein goes and in turn binds in the cytoplasm, and the E2F again becomes active. And the activation of E2F results in the activation of the genes which are important to initiate G1 to S-phase transition. And one of the important genes which is initiated is cyclin E. So this retinoblastoma gene has been found mutated or altered in range of different cancers. So the first is inactivation of this retinoblastoma gene by mutation, which happens specifically in retinoblastoma, osteosarcoma, as well as small cell lung carcinoma. And there is also a methylation of retinoblastoma gene that resulted in its complete reparation, and that has been found in brain tumors as well as the diverse type of tumors. In the third, the retinoblastoma protein has also been sequestered by ID1 and ID2 proteins, and also is been sequestered by the E7 viral oncoproteins. And that has resulted in many different cancers as well as neuroblastoma, melanoma, as well as cervical carcinoma. And second important cell cycle components are cyclin. We have seen that the cyclin undergoes a cyclical changes during cell cycle progression. So there are different cyclins which are important to initiate and regulate the cell cycle progression. So cyclin D, which is composed of different types, that is cyclin D1, D2, and D3, is important to initiate the entry from the restrictive point. And then cyclin E, which is important to traverse the cell between G1 to S-phase transition. So this cyclin D has also been found overexpressed through different tumors. And the overexpression mechanisms are quite different between these tumors. So in the first case, cyclin D1 overexpression is normally through an amplification of cyclin D1 gene. And that has been found with the breast carcinoma and leukemias. And second type of overexpression has also been observed by the hyperactivity of the cyclin D1 gene at the promoter level. And that is in turn catalyzed by different mitogenic pathways. Also, there is no degradation of cyclin D1, and because of this, the cyclin D1 also gets accumulated, and that has been found in wide range of tumors. And thirdly, the cyclin D3 overexpression is also brought about by hyperactivity of cyclin D3 genes. And that has been observed in case of hematopoietic malignancies. And another cyclin which gets overexpressed is the cyclin E, and that is typically observed in breast cancers. So what happens when these cyclins actually gets overexpressed? So this overexpression or amplification of the copy numbers of cyclin can be detected by using FISH. So this FISH is nothing but a fluorescence in situ hybridization. So here are the head and neck squamous cell carcinoma cells. And here the blue color are the nuclei, and the blue arrow here depicts a copy of this cyclin gene. And here the cyclin is cyclin D1. As you can see here with the blue arrow, there is only a single copy of gene which is present, as seen by a single dot. However, in other carcinoma cells, you can see here, which are marked with the red arrow, you can see alteration of this cyclin D1 genes, which are present multiple copies. In some cells, they are present in 3, or some cells they are present in more than 5 copies. And because of this cyclin D1 overexpression, that results in the loss of control over retinoblastoma protein. And we have seen that this retinoblastoma is very important for the cell cycle transition between G1 to S phase. So once the cell loses a control over this retinoblastoma protein, then there is an aberrant cell cycle progression which result mostly into form of cancers. And what happens exactly when we overexpress or when the cyclins gets overexpressed? So this is an experiment which was performed with the normal cell as well as with the breast cancer cells which have elevated cyclin E levels. And they are stained by the red protein, that is centrosomes, and the greens are the microtubules, and the blue are nothing but chromosomes. As you can see here, in the wild type cell, the chromosomes are equatorially aligned, which are nothing but in a metaphase of the cell, where there are centrosomes, which results in formation of the bipolar spindles. But on the other hand, if you look at closely at the breast cancer cells, there are huge number of centrosome amplification which has resulted in radiating microtubule from every possible direction. So such a huge level of catastrophe can happen if you just overexpress a single cell cycle component. And in this case, it's a cyclin E. And because of this, microtubules which are aberrantly formed within a cell nucleus, that result in the change in the chromosome number as well. As you can see, the blue color of the chromosome get distributed throughout, and it's not aligned in a proper way. So this can create a lot of havoc within a cell, and finally, this aberrant regulation of the cell cycle culminates in tumorigenesis. So this is one of the very important stuff which we should control. The third protein which often found mutated is p53. And in my previous slide, we have seen that the p53 gets activated because of the phosphorylation by CHEK1 and the CHEK2 protein, and this happens due to ATM and ATI kinases. So the phosphorylation of one of the inhibitor of p53, that is MDM2, results in its activation, and this activated p53, which is a transcription factor, goes and binds to the promoters of wide range of genes. And one of the important gene which it activates is p21, and which is a CYP family of inhibitor which inhibits the cyclin CDKs, which are important for the G1 to S phase transition as well as during the S phase progression. So this is the sample which is taken from a lot of tumor patients, which are wide variety of tumors. So these include ovary, colorectum, head and neck cancers, and different types of cancer, as you can see here on the y-axis. And on the x-axis here represents the percentage of tumors with the change in the p53 sequence, that is p53 mutations. And what we can see here, majority of the cancer have accumulated with this p53 mutation. So from this bar graph, you can understand the importance of p53. If there is a change in the p53 sequence, and that we called as mutations, if this happens in a cell, that results in wide range of tumor formation. And that ranges from cervix till the ovary cancers. So with this, I would like to end my second part and also like to conclude the cell cycle seminar by giving you the summary. And what we have seen here in the first part is the S phase progression, and with the help of the S phase CDKs, which activates MCM helicase, and that results in the bidirectional replication, and during the onset of G2, there is completion of DNA replication. Following that, we have also seen about mitotic protein kinases, which are cyclin B, CDK1, aurora kinase, and polo-like kinase that work in a feedback loop and activate each other, which carry out a variety of transactions during the process of mitosis. And that includes the chromosome condensation, nuclear envelope breakdown, and fragmentation of Golgi apparatus, as well as the spindle formation, which is because of phosphorylation of the centrosomes. And we have also seen the duplication of centrosomes, which are important to radiate these microtubules, which in turn attaches itself at the centromeric site with the help of huge proteinaceous structure that is Kinetochore, and that results in the segregation of the sister-chromatids. Finally, we have also seen about a comparative overview between a meiosis and the mitosis, and how exactly the meiotic chromosomes are aligned as compared to the mitotic chromosomes. And we also looked into different checkpoints which operates, and that to a DNA damage checkpoint which gets activated because of the DNA damage, and with the help of ATR and ATM kinase that results in the progression or the smooth transition of the cell cycle. And we have also seen that any defect in the cell cycle component can lead to tumorigenesis. So with this, I would like to end my seminar, and these are the references which I have used to create the cell cycle seminar series, which I have divided into the part 1 and part 2. So the book by Weinberg, and there's a book by Lodish, as well as Bruce Alberts, and there is a very nice book on the concepts of genetics by Klug and Cummings. And finally, you can also have a look at the cell cycle or molecular approach, a book by Cooper. So thank you all.